In this video, we will discuss about effect of different CAR construct on iPSC differentiation into T cell lineage. iPSC is this induced pluripotent stem cells. It can be differentiated into different kind of cell lineage, and so the idea is to transduce this iPSC with the CAR construct so that it can differentiate into the T cell lineage. So they start with different kind of CARs from first generations to third generations, and first generation that does not have the co-stimulatory domain, and in the case of the second generations they have the co-stimulatory domain of this 411B, and in the case of third generations they have two co-stimulatory domains, and for the control they don't have the antigens, and for the mock they don't have any CAR construct, and they also check it for the inducible 28 zeta, that we will discuss why they use these things. So first, in order to check the different CAR construct, the methodology that they used is first they prepare the viral lentivirus and then use the viral transductions at day 0, and at day 24 they check the signals, and for the mature T cell lineage they will have this CD5 and CD7 double positive cells, and this they are able to see a high in the case of in the case of this 1928 beta Z, but they are low in the case of 19 zeta first generations and the second generations with 1928 zeta. And for the immature T cell lineage, double positive, the marker says CD4 plus and CD8 plus double positive cells, and they are able to see, and these signals are also low for the case of first. These signals are also low for the case where they use first generation CAR and the second generation CAR with 2028, indicating that the transduction of CAR might impair the differentiation of iPSC towards T cell lineage. So they doubt this in the case of 1928. There is a tonic signaling that is triggered due to non-specific activation of CD3Z. So in order to check whether the differentiation loss in differentiation or impaired differentiation is because of CAR transductions, they prepared a inducible anti-28 zeta where they use doxycycline to induce the translation of this CAR, and they are able to find out the same results, that with the inductions of this 1928 zeta they are able to see reductions in the T cell lineage populations, and rather populations that is more aligned with the NK cells seems to be formed. So in order to the hypothesis that they developed is that the impaired differentiation may be because of non-specific activation of CD3 zeta. So in order to check whether CD3 zeta is activated in the case of second generation CAR, they took this CAR construct and induced the 1928 zeta with TGFR, and after 14 to 16 days they check the signals again, and they are able to see CD3 plus zeta signals increased. Phosphorylated CD3 plus zeta signal increased in the case of 1928 zeta, but they are not increased in the case of 19BB zeta, indicating that the non-specific tonic signalings are being observed in the case of 1928 zeta CAR transductions, and that is causing or impair the differentiations of these iPSCs to T cell lineage. And these things they have shown in the flow cyto. One of the representative flow cyto image are shown in the panel D, where we can see that in the case of 1928 zeta the CD8 beta positive and CD4 positive cells are very low, around 10%. It is decreased compared to the control, and in the case when 1928 BB zeta is used, they are able to see almost similar amount of CD8 beta and CD4 positive populations, indicating that 1928 BBZ doesn't impair the differentiation of iPSCs to T cell lineage. Rather, this 1928 zeta impaired the differentiation of iPSCs to T cell lineage. We will discuss further results of this paper in our subsequent video. I hope you understood so far how they come up to this point that 1928 zeta is 1928 zeta impaired iPSC differentiations to T cell lineage, but not 1920 BBC. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.